All right, last but not least, we have Jing Zhou from McGill University, who will speak to us about longitudinal iron in caudate related to control network connectivity and planning in older adults.
Okay. Hello, everyone. My name is Jing Zhou, a PhD candidate in the lab of brain and cognition directed by Professor Nathan Spring in the McGill University. And today, I would present the longitudinal ion accumulation in the head caudate related to frontoparietal control network connectivity and executive function in older adults. Sorry. As we all know, the iron is an essential trace element in the human body, and the iron deficiency can lead to anemia. But what's the mechanism of action of iron in the human body, especially in the brain, and why does it matter? There are two categories of iron in the brain, and one is heme iron, and other one is non-heme iron, which we focus here. The non-heme iron includes the ferritin for the iron transport and ferritin for the iron storage. And the non-heme iron involves in many brain neural functioning, including the mitochondrial respiration and ATP synthesis and neurotransmitter synthesis. However, as people age, the iron deposit in different areas of brain may impair the brain function and cognition because, because non-heme iron is a very active oxidizer. Its accumulation outside of binding complexes investigate proliferation of reactive oxygen species and induces oxidative stress. So how to measure brain iron? The MRI methods such as the ultrastar and quantitative split mapping, the QSM, are widely used to quantify the iron content. The QSM approaches, because of its high spatial resolution and high sensitivity and high accuracy, are able to measure tissue iron content based on GI phase images. Just as shown in the figure, in the basal ganglia, it's really quite bright on the QSM images. So what does it mean? A previous post-mortem QSM study has demonstrated a stronger positive linear correlation between the chemical iron concentration and bulk susceptibility. So the great QSM values imply the greater iron concentration in tissue, and the basal ganglia structures have the largest iron concentration and increase with age. And next, I would introduce how the iron deposition affects the healthy aging. The striatal, as important part of the basal ganglia, has been the focus of research in the past, and it includes the caudate and the putamen. Is one of the most iron-rich regions in the brain. And previous QSM studies demonstrate age-related increase in the striatal iron levels, and more cross-sectional studies report that the high iron levels in the striatal, especially for the caudate, were related to volumetric atrophy and reduced the frontal striatal activation and intrinsic coherence with the striatal, as well as the decline in cognitive performance. Here, we focus on the head or caudate, where the dopamine depression is greatest and is heavily connected with dorsolateral prefrontal cortex, and support the successful executive goal-directed cognition. So the aims in this study, we first examine the impact of the longitudinal iron accumulation in the head or caudate on resting state function connectivity changes in the frontoparietal control network, and then examine the relationship to the goal-directed cognition, the spatial planning. So in the current longitudinal multimodal study, 130 cognitive unimpaired old adults at family risk for AD were included. And we corrected the QSM and resting state function connectivity at baseline and follow-up. And most of participants also completed the special prenatal test as well as assessment of AD risk factors, including the tau PET and APOE4. Next, let's look at the imaging analysis and post-processing involved in this study, and these 4 modules in total. So first of all, for the QSM processing, the original magnitude and phase images were combined to get offset corrected magnitude and phase images. And then these corrected images were submitted to TGV pipeline to do QSM reconstruction, and then we got QSM map. And then next, to obtain the head caudate RI in QSM space, we combine the caudate RI generated by free-surf segmentation and the caudate subfields RI generated by a fMRI-based subcortical analysis. As for the resting state fMRI data processing, the both data were extracted from each participant's GPIB solution from the parcels comprising the 7 networks. And then the functional connectivity matrix were created by computing the product moment correlation coefficient between each pair's origin. And finally, we use the 2-condition behavioral partial least squares to examine the relationship between the longitudinal change in ion content and the frontoparietal control network. So what we found, first of all, we observed that ion levels in the head of caudate increased over time, but there's no changes in the frontoparietal control network. And more interestingly, we found that increasing ion content in the head of caudate was significantly associated with longitudinal change in frontoparietal control network, with both increases and decreases in frontoparietal control network. And in terms of the association with executive function, we observed the prenatal score decreased over time. And more importantly, we found that the declining prenatal was associated with the brain connectivity score derived from behavior pairs using the linear model. And further, to examine the association with the age-related factors, firstly, we use the partial correlation analysis to examine the association with the tau PET. And there's no association between the tau PET and the head caudate ion and the brain connectivity score and the special prenatal score. And then to assess the potential moderating role of the APOE4 on the relationship between the ion content and brain connectivity score and the special prenatal score, we use the linear mixed model again. And firstly, there is no difference between the APOE4 positive and negative groups for the all measures at baseline and follow-up. And again, there is no significant results was found for the linear model analysis. And in general, as predicted, the ion levels in the head of caudate increased over time, and the accumulation of ion occurs with changes in brain function, and this interaction covariance executive function decline in all the adults, and there's no association were observed between the age risk factors like APOE4 status or PET tau levels. So these findings suggest that the longitudinal interaction between the ion and the brain function are independent of the age pathology, suggesting a unique source of age-dependent brain and cognitive decline. And finally, thank you for your attention. Thank you, my supervisor and all lab members and all collaborators. And if you have any question, please come to my poster on Wednesday and Thursday. Thank you so much.
Okay, there's a question from the app. The increase in spatial resolution and high sensitivity might increase noise levels. How confidently can you eliminate the contributor noise in ion level estimations?
You mean the noisy for the quantity for the QSM in the measured QSM?
I'm not sure. I think so.
Okay. So, yes, that's a quick question. So, normally for the QSM processing, and we're using the TGY pipeline, and this pipeline combined the QSM may process in the 3 steps, like the first step, the phase unwrapping, and background remove, and the field to sensibility inversion. Just combine the 3 steps. The QSM may processing steps into one iteration, so that can reduce the noise. Yeah.
Thank you very much.
Thank you. Thank you so much.
All right. Thank you all very much for a lovely session, and please join me in thanking all the speakers.
Thank you for staying till the very end of the last session. Thank you.